Hi students, this is the second part of the lecture regarding with the structure of the heart, and hopefully you would have liked the first one. Let's jump to the. So there are two types of cell in the heart. One is called contractile cells, and others are called non-contractile cells. Contractile cells, as the name indicates, that they are related with the contraction of the heart, and they are responsible for the mechanical events of the heart. Mechanical events mean contraction, and relaxation. More specifically, contraction is called systole or systole, and relaxation is called diastole or diastole. So these contractile cells are called cardiomyocytes. Cardio mean heart, and myo mean muscles, and cyte mean cells. So these cardiomyocytes are related with the contraction of the heart, and these are the muscle cells. They are elongated and striated. If you can see the striation, and they contain two nuclei. They are elongated and branched. And the group of cells that contain more than one nuclei are called syncytium. So these are, if these cells are present in the ventricle, so these group of cells will be called ventricular syncytium, ventricular syncytium, and the cell that are present in the atrium is called atrial syncytium. Let me erase it and move forward. And these cells do not have they do not need any external stimulus. To contract, as smooth muscles need external stimulus to contract and relax, but these cells need stimulus from inside of the heart, and that stimulus is called action potential. How this action potential works, let's see. These cardiomyocytes actually have filaments of actin and myosin protein. So suppose these are thin filaments and we call it actin filaments. And these actin filaments are connected with a disc that is called Z-disc. Z-disc is made up of special type of protein that is called actinin. Actinin. So these filaments, let me make another side. These thin filaments are called actin filaments, and due to the space, I have because they are made up of actin protein, and in the center there are thick filaments. These thick filaments are made up of myosin protein. That is why they are called myosin filament. Myosin filaments. These are actin filaments. So when electric impulses excite, and this the whole unit is called sarcomere. This whole unit is called sarcomere. So when electric impulses are action potential, excite this sarcomere, so these actin filaments slide over the myosin filaments, and the size of the muscle is reduced, and in other words the muscle contracts. And when these filaments move away and they slide away from these myosin filaments, then we say that heart is relaxed, the muscle is relaxing. So cardiomyocytes are made up of actin and myosin filaments. Myosin filament are the thick filaments, and actin filaments are the thin filaments. And there are some types of protein that is attached to the actin filaments that is called troponin. And when there is injury to the cell, this troponin spills into the blood. And the level of troponin in the blood is used as a biomarker for cardiac injury. So cardiomyocytes contain different types of protein like actin, myosin, and troponin, and contains sarcomeres. And these sarcomeres are contained another another structure that is called myofibrils. So myofibrils are basically the cylindrical shaped fibers that contain sarcomeres, and some proteins like troponins are also attached to these actin filaments. So the basic unit of contraction in cardiomyocytes are the myofibrils. Myofibrils. And myofibrils contain sarcomere. This unit is called sarcomere. Cardiomyocytes are connected with small discs. These discs are called intercalated discs. Intercalated discs. We will discuss it in detail when we will discuss the action potential of the heart. Okay, let's move to the non-contractile cells of the. They are very interesting cells. There are different kinds of non-contractile cells in the heart. So one is SA node. SA node, S stands for sinoatrial node, and it's also called the natural pacemaker of the heart. Why it is called the pacemaker? Here is SA node, and it is located just below the superior vena cava. Superior vena cava in the right atrium. Right atrium. So these cells have lost their ability to contract, and now they have taken a new function that is called production of action potential or production of electrical impulses. The second non-contractile cell is called AV node. AV node mean atrioventricular node because it is present at the junction of atrium and ventricle. That's why it is called atrioventricular node. So these SA node produce electrical impulses, and these electrical impulses excite cardiomyocytes, the contractile cells of the heart, to contract. And this action potential is related to the electrical events of the heart. It performs the electrical events of the heart. What are those electrical events that are called depolarization, polarization, and repolarization? Polarization. What is depolarization and repolarization? A cardiomyocyte is usually non-polarized. There is equal amount of positive charge and negative charge on these cardiomyocytes. But when these impulses reaches and excite these cells, and the distribution of these positive and negative charge are disturbed by the influx of calcium ions, and they become more positive. And this process is called polarization. And when a cell is polarized, now it is ready to start for the contraction. And after contraction is done, the process is reversed, and it becomes again neutral, and this phenomena is called repolarization. And other cells that are non-contractile are bundle of His. This is bundle of His. And the bundle of His is divided into the left bundle branch and right bundle branch. And these bundle branches again divide into small branches. These small branches reach to the endocardium, and these branches is called Purkinje fibers. Purkinje fibers. So you can say that that SA node 1, AV node 2, bundle of His 3, bundle branches 4, and Purkinje fiber 5. There are 5 types of non-contractile cells in the heart, and they are related with the electrical events of the heart, that is depolarization and polarization. And in other words, depolarization means after that the excitation of the cell, and after that the cardiomyocytes are ready to contract. And repolarization means relaxation of the the cells. So there are almost 300 billion cardiomyocytes in the cell, and there are only 5 contractile non-contractile cells in the heart. So SA node is called the natural pacemaker, and AV node is atrioventricular node. It also has another function, that when impulses are generated by the AV node, SA node, sorry, it stops them for 0.1 second. This 0.1 second is called refractory period. Refractory period. And AV node does not let these impulses to move through bundle of His until 0.1 second is passed, because during this period atrium contracts and blood enters into the ventricle, and they are fully filled. if if it doesn't hinders their propagation, and impulses are directly passed into the ventricle, so ventricle also starts contracting along with atrium, and there will be a chaotic situation. So AV node is also called refractory node. We have already discussed about syncytium, that what is atrial syncytium and ventricular syncytium. These are actually the cardiomyocytes. Why they are called syncytium? Because they contain more than one nucleus. That is why they are called syncytium. So there is a unique structure in the right ventricle that is called moderator band. So this is only present in the right ventricle. In an echocardiography, if we did the apical four-chamber view of the heart, so moderator bands are visible, and they help to distinguish between right ventricle and left ventricle. So the moderator band, remember, is present in the right ventricle. So what is moderator band? Moderator band connects interventricular septum to the anterior papillary muscles. So here are the papillary muscles and chordae tendineae. So anteroventricular septum, and here is the moderator band that connect interventricular septum to the anterior papillary muscle. This is also called septomarginal trabecula. Trabecula, I told you that it is a column, and actually this is type of the muscle band, and that is derived from interventricular septum and connects with the anterior papillary muscles of the right ventricle. So there are some other structures that are present in the atrium and ventricle. One is pectinate muscles. Pectinate muscles are actually the folds, and they resemble with the teeth or the comb. Resemble with the teeth or the comb. And the main function is, it is considered that they increase the power of contraction of the right atrium. And pectinate muscle of the atria are different from the trabeculae carni. And we discussed that the trabeculae carni, trabeculae means the column, and carni means meat. So these are the meaty columns that is present in the ventricles, and I showed you in the previous lecture. They are present in the at the inner wall of the both ventricles. And these pectinate muscles not only increase the contraction of the atrium, but also reserve the volume during severe stress, volume of blood. In ventricle, trabeculae carni, we discuss that these are the meaty ridges or meaty columns, and they are present in both right and left ventricular, and they are different from pectinate muscle that are present in the right atrium or atria, and their function is the function of the trabeculae carni is similar to the function of the papillary muscles. And you know that the function of the muscle is to contract. And papillary muscle, we also discussed that papillary muscles are nipple-like structures. That is why they are called papillary muscles. So here is a moderator band, and this is interventricular septum, and it is originating from interventricular septum and connecting the papillary muscles. And you see these are the strings of the papillary muscles that are further attached to the cusp of the valve. So it is like a bridge between interventricular septum and the papillary muscles. And in echocardiography, it is seen as a bright dot in the right ventricle, and it is a landmark to recognize, to differentiate between right ventricle and left ventricle. We discussed cardiomyocytes, that cardiomyocytes are two types of cells, contractiles. They are related with the mechanical events. Mechanical events mean systole and distole, and they get their stimulus from sinoatrial nodes and atrioventricular node. And their main objective is to produce force of contraction, and they are connected with each other by intercalated discs. This is one cardiomyocytes, the other cardiomyocytes, and between these there is an intercalated disc. And they receives action potential from SA and AV node, and actually the action potential is their stimulus to contract. And non-contractile, these are specialized type of the myocytes. They have no ability of contraction, and they are related with the electrical events of polarization and repolarization of the cardiomyocytes. And polarization mean contraction, and repolarization mean relaxation. And this process is spontaneous and automatic. So these non-contractile cells automatically produce these impulses, these action potentials. So this is called autorhythmicity. And these non-contractile cells need no external stimulus, and plural is stimuli, while cardiomyocytes external stimulus is action potential. And SA node or sinoatrial node, that is a non-contractile cell, is also called the pacemaker or Keith-Fleck node, node of Keith-Fleck. So these are the different names of SA node. And how they are able to produce electrical impulses, they have funny channels. Funny channels by which influx of the calcium ion takes place and impulses are produced. So atrioventricular node is also called a refractory node, and it delays the electrical impulses generated from SA node. So we will discuss in detail that how action potential is produced in SA node by the funny channels. And AV node is also called node of Tara.